announcement. So if yesterday, if yesterday, rather than Monday, I recorded a presentation which you may or may not have. It was a completion of the cell stuff. So I finished that stuff about the resting membrane potential, a little bit about exocytosis and receptor mediated endocytosis, and then I did the cell cycle stuff, which I think is important. I didn't go crazy with all reviewing how assembly of DNA and RNA and replication forks and bubbles. You all had that. I don't want to revisit it. That's not my area of expertise. We have someone who is. You all have been subjected to that torture. Sure. Nevertheless, we are going on to the hard stuff. That was the introductory stuff. So listen to that because certainly there's going to be questions from that chapter, what I call chapter 3 of cell material on the exam. The next unit I have loaded, there's 4 PowerPoints. The textbook that we get from the book bundle textbook is on there as well. That's the Welsh Hall textbook, wherever I put it. It'll be, it's their chapter 9, similar, and we have 9A, B, and C. So they both have to do, and these are effectively muscle physiology, which means this is the start of nerve and muscle physiology. So this is what we're getting used to. Let me make sure I started because I keep forgetting. Yeah, I've got the red dot, so I am recording. Oh, I feel better. So you'll find that here on the blackboard. I endeavored to take care of that. I am on my game, more or less. You're looking at your watch already? What did I do? I've been talking for 2 minutes. Not even a minute. No one has that short of an attention span. Even my kids don't have that short of an attention span. And they tune out, as you know, tuning out parents is easy. Not that any of you would do that, of course. I'm confident of that. WTF. Okay, they want to center picture in picture so you can use it on. Just take a chill pill. Voila. So this is both. The beauty of this is that we're doing effectively neuromuscular physiology. The muscles that we're dealing with principally in this chapter, even though we will touch on smooth muscles, we'll save cardiac muscle physiology to the heart unit, are principally skeletal and smooth. And so we spend a lot of time on skeletal. There are outstanding animations, 3 of them in this particular, and it's why if you're going to download it, it's a pretty large download. However you view it, you can view it. It's one of those things that you can't fit because there's limited space on Ultra. And so as you can see, it's really important because now we're going to see the nerve component and the muscular component. So it's multiple areas. We have to look at the neuromuscular junction, how the signal comes from the nerve, goes to the muscle, how it triggers it, how specifically it triggers it, what's involved with the various kinds of channels that are involved, then effectively how we transduce or transform the energy from one form to another. From electrical energy, we're starting with chemical energy, we're going to be going to electrical energy, then we're going to be transforming it to mechanical energy. And so we call it excitation, contraction, coupling. So it's a variety of changes, if you will, just basically modifying or changing the form of energy might be the better way to express it. And then after that, we have to look specifically at the mechanism, which is called the cross-bridge cycle. You may have looked at it previously. It's well worth looking at it. Just thought I'd bring that in and get everybody up to speed on that. So half the body's mass, well, our muscles, most of our weight comes from our muscles more than any portion of the structure, certainly compared to, people always think, well, your bones are really heavy, and it's really not. It's more about the muscles than anything else, and the ability to transform or change chemical energy into mechanical energy. So you're going to see. And so all of that is important. We take a look at the general anatomy of muscle tissue, which you may or may not. We talked a little bit about that. I'll go over it. So we look, and I don't see, as you remember, in advanced anatomy, I don't love teaching muscles. I hate playing name that muscle. It just doesn't do it for me. If you're going into field where you're going to have to learn those muscles, they're going to teach the ones that they need you to know. So we're going to look mostly at how they function and what their characteristics are. From a we use different terms. When we get into the cellular level, we're going to spend most of our time. Sarco is the prefix. We don't have a cell membrane, we have a sarcolemma. We don't have cytoplasm, we have sarcoplasm. Okay, we don't have an endoplasmic reticulum, we have a sarcoplasmic reticulum. I know they're all the same, but we're using a different prefix. When we're using myo and meso, we typically, myo and meso, we're looking for larger structures, some macroscopic, some microscopic. So all of that's important. 3 kinds of muscle, you're already familiar with this, I'm sure. The ones that is voluntary, skeletal muscle. Another name for skeletal is striated muscle because we can see the striped appearance, particularly microscopically. Voluntary control. Cardiac is the interesting blend. It's striated, though not as distinctly, and it's not under voluntary control. So it's like a blend, smooth, where you can't see the striations, which is involuntary, and cardiac, which is sort of a blend. It's that unique thing about endurance and strength matching together, the way I framed it, and I always do. So that's neat, and we really will spend a fair amount of time looking at the heart phys, which we really didn't do last time. We just basically did the basic here's the chambers, heart, and all that good stuff. So we're getting there, and so there we call them fibers. That's another name for the cell. So terminology is important. This is where people trying to say the right word mess up. So I was going to say something else. She's looking at me. You're going to drop an F-bomb, weren't you? This is where, oh God, I can see Abby and she's smiling. You look nice today. You look nice. You all look nice, but I got to tease her. And after full disclosure, it was so darn cold yesterday. I broke down when we were shoveling out the cars and getting the driveway. I got the long johns out. I'm not ashamed. I got layers here. I got a sweater under this. I mean, I'm layered to beat the band. Just saying, full disclosure. So when we use terminology is important. You're going to hear about fibers, the cell itself. You're going to hear about fibrils. You're going to hear about filaments, progressively smaller. And it's easy, and I'll get them mixed up, but those are really important terms test-wise, because those are easy ways to make students get the wrong answer, to use that kind of term. I don't like to be terminology dependent, but that's a look out for. My tests or other people's tests. So skeletal muscle tissue, very unique. Bones and skin, yeah. Fibers are the longest of all muscle fibers. They can be as long as roughly a foot, more or less. Okay, the longest muscle is, maybe you don't remember we talked about the sartorius. It's a muscle that basically goes from your hip area, actually from the anterior superior spine of the ilium. Put your hands on your hips right up there, all the way down to just slightly below your knee. It's a linear muscle, and that is the longest of the muscles. So the fibers are quite long. You're going to see when we talk about nerve cells, they can be even 3 times that long. They can be a meter that are there. So we call it voluntary, conscious control, contract rapidly. They tire easily, but we replenish them very, very rapidly. I mean, until fatigue sets in, imagine how rapidly you can contract that muscle over a period of time, depending on the kind of work that you're doing. So again, skeletal, striated, involuntary, cardiac, and the heart, we'll get to it. Again, the combination is interesting because I really think this is that unique muscle. It's so powerful, yet it's constantly beating. We'll get to that. And smooth, really, this is the unit in physiology where we really explore smooth muscle. We were maybe talking about peristalsis and these hollow tubular structures that move fluids and masses along. This is where we begin to understand the mechanics of how they can do this remarkable degree of endurance when they're strong, but by no means nothing compared. So all of those things, whether it's a respiratory passageway, a blood vessel, a genitourinary passageway, or a GI passageway. Okay, and visceral is a good term. Again, I like non-striated to me because you can't see the striations. Doesn't mean they're not there microscopically. And here's some examples, nothing I would ever ask you about, as you know. The big deal here, that whole resting membrane potential, what makes them excitable, okay, is the fact that they can respond to stimuli. They can respond to mechanical stimuli, such as reflex. But here we're going to be looking at a variety of different stimuli, particularly the events at the neuromuscular junction. They contract, which means they shorten. Okay, it's not a folding thing. Remember, with the term, I'm sure you're familiar with it a little bit. This is where we really understand it. The sliding filament theory. Remember, filaments are they get smaller. Fibers, fibrils, filaments slide over one another, so they appear to shorten. I beg your pardon. They do shorten. They appear to contract, depending on the arrangement of the muscle. That's when you go back and look at the anatomy of the muscle. Do they bulge in the middle like a biceps, or your biceps? Or mine kind of goes down. One freaking smile. You're not being helpful. I've missed talking to real living people in a group, teaching wise. Sitting there and talking to my computer. The dog looks at me and goes, WTF are you doing? Stop doing that stuff and give me a biscuit. Okay. And they can stretch and they're elastic and dot, 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 dot. So movement, by that, walking, digestion, pumping blood, posture and stability, stabilization, generating heat when we shiver, something we should be very familiar with given the climate that we are experiencing today. Yikes. And tomorrow, and the next day, and for the next week. Oh. Okay, here's the organization that's so important. When we're talking about muscles and nerves and the heart, we're talking about extraordinarily good blood supplies. The big problem is that, again, these are the highly differentiated cells. When they're damaged, we do not get repair. Okay, we form scar tissue. They cannot replace themselves for all intents and purposes. We look at the sheets and the attachment. This is a little review of the anatomy. What we call the neurovascular bundle, a term that you should be familiar with. What's interesting with that, when we looked at bone, you might remember, the blood supply entered the bone and then made its way through the interior. Here, the blood supply is coming from without. It's from the exterior, so to speak. So we're not seeing something penetrating and being interior. We obviously have conscious controls. We require a ton of oxygen and a ton of nutrients. And because we use them so frequently and so vigorously, for the most part, there's a lot of waste product removal that has to go on. It's a really interesting system. The big problem when you get fatigue is less about absence of ATP. It's more about accumulation of waste products. When we see someone cramp up in a sporting event, you see it all the time. Most of the time, we're talking about accumulation of waste products more than anything else. And it's not just lactic acid. There's a whole lot more about electrolytic imbalances and things like that go on with this. It's very interesting. This sort of training for people who are going in, who are interested in athletic training and conditioning, and many of you participate in sport. I mean, there's a lot going on here with that as well. And I mean, it may have changed. I would certainly never attempt to take the place of or present information and say this is what you should be doing to rehabilitate this or that, unless it's something I have very, very specific knowledge of. And the state of training has changed dramatically over the years. So I just want to frame it as that. Look, I haven't treated a patient in more than a quarter of a century now. So, I can tell you what we used to do, and I have an idea what's being done today, but by no means an authority whatsoever. So moving on. So again, we have this idea of layering and arrangements, and nerves are very similar. The interesting thing about nerve is the blood supply is right down the middle of it. It's on the interior. And so you're going to take a look at it and you'll see the arrangements of nerves, similar language, except you're going to see neurium rather than mesium. Epimesium is that dense, irregular material that's around. It's that sheath. Some people call it silver skin. When you're looking at, if you buy a big chunk of meat and you're trying to cooking it, something you typically would remove or trim off because it gets gnarled and irregular, and it's definitely not good eats. Okay? So that's something, if you're into getting large chunks and doing some of your own butchering, you can do that. For you newbies in here, I'm a foodie. We make, and it's such an easy recipe. We morphed this great potato soup recipe into clam chowder. Bang. Oh, so we cook. Were you going out to the store on Sunday afternoon in the middle of the snowstorm? I don't think so. Oh, my recipe. Oh, just saying. Last night, my wife got into a. What do you call they? What did they call them? What are they, peanut butter blossoms. I call them kiss cookies. You know, we got to we got to make those tonight. Okay, I can do this. Get that big stand mixer out and so you're going to see different layers. Easy for me to show you. Here's the exterior. The continuation of that covers tendon. Okay, it becomes a sheath of the tendon. Tendons attached to bone. Sometimes it'll go directly into bone. Tendons are an indirect attachment, and anatomically speaking, and if it goes directly into the bone, it's called a direct attachment. Again, that's anatomy, not physiology, so I'm staying away from it. And you can see when we're looking cross section, when you go to wherever you buy, Giant Eagle, Walmart, wherever your butcher shop, they cut these things tangentially, so they're basically cross sections. And the different cuts refer to the different cross section. And when you see the fascicles, they are surrounded by a less dense connective tissue with an increasing amount of fat. That is referred to in the trade as a marbling fat. The more you have of that, okay, the better quality the meat is. So that's where you see your grades. Typically in the store, you get at least choice, or if you want to pay, a very large, you have prime. Or if you're going out to a really fancy restaurant, they're going to serve you prime. You're going to pay. So just to give you, most of the time, CAB, certified Angus beef, typically is choice, unless you're paying extra for prime cuts. And I'm speaking to all of you who are carnivores and omnivores, but like I said, I have tons of vegans and vegetarians in my family. I'm good with it. You want to do plant-based? That's your call. So each of these, the perimysium, create these little bundles we call fascicles. If you pull them out, here's the perimysium, basically the border, and now we have the endomysium, okay, which is the membrane, very delicate, loose areolar connective tissue surrounding an individual fiber. That's a cell. It's a unique cell because it's multinucleated. It's a unique cell because all it really is a fusion of multiple cells, and it's filled, as you can see here, with rods of protein. It's nearly all protein. There's some blood vessels, there's some membranes, there's some other stuff in there, sure. Some fat in there, sure. But effectively, the individual fiber is this very interesting thing. It's got the endomysium around it, a external sort of boundary, and then deep to that, you will now get to the next prefix where you're going to see the sarcolemma. It's really important when we get into what triggered, what happens with the sarcolemma, what are T tubules, okay? What's the triad system is there? It's a very unique system to become familiar with. So here you can see it, the fibers, the fibrils themselves there. I did it. And those rods, and we're going to pull those out, and we're going to see the filaments, and that's where you're really going to get a good look at the striations and what makes them. And so here's insertion. We're not doing that. Directly into the bone itself, or the area around the cartilage. Typically, it's more bone, maybe a tiny amount of cartilage. And indirect tendon, or what's called, and you might remember this from anatomy, aponeuroses. So that linea alba, the white fascial tissue, that's an aponeuroses when someone has the definition we call a six-pack. Or here, the Galea aponeurotica, which you have the frontalis and the occipitalis muscles, so you can do the whole brow wrinkling thing. All of those. So there's a lot of, and you'll see that if you just go back and look at the anatomy, all of those gray areas are that. So that's a relatively common, the plantar fascia, very, very frequently injured. When people get heel spurs, it's typically the spur is inconsequential. It's tearing of the fascia at its attachment inferiorly to the calcaneus more than anything else. That I actually know something about. So voila, epimysium, fascicles, perimysium around the individual fascicles, bundled we see fibers, and as you can see, they're surrounded by that lucioareolar endomysium, and underneath it is the sarcolemma. And what we pull out of there, and you can begin to see the banding pattern that's there, that alternating light and dark areas. Now we have myofibrils. And thankfully, they're all identical. And they're broken down into tiny units called sarcomeres, as you will see. Each of these are little blue lines. We blow this up, magnify it so you'll be able to see it in the subsequent slides. And so they're all identical. They use the analogy, and any analogy works, like boxcars. So they're identical boxcars. If we study one, they all do the same thing in a particular thing we call a motor unit. So they all contract simultaneously and effectively. It's like a room like this. It's sort of the reverse. If you go to Disney and you have like in the Haunted Mansion, you have the stretch rooms. These, it's like a bad sci-fi movie. You trigger something and it turns. Indiana Jones, Temple of Doom. Saw that? Thank God. You're one of us. There's a great two-part series, everybody, just dropped on HBO, part one, about Mel Brooks, the 99-year-old man. For you who don't know me, I'm a Mel Brooks devotee. He'll be at 100, Lord willing, on the 26th of June. And he's going to make Spaceballs Part 2, the movie. It's a great biography. You just laugh out loud. Just really. So here we are. The skeletal muscles, long cylindrical cells, multiple nuclei. That's why they're not, you can't replicate these. Muscle-fiber plasma membrane, by that. Cytoplasm, sarcoplasm, contains, understand, the demands are tremendous. It's got to have a ready energy source. So there's only two areas in your body you can store glycogen. We have very limited carbohydrate storage. Excess carbohydrate gets stored as fat. I'm an authority on that. So much candy, so little time. To make my point, on my way here, I visited the Candy Man. You know, not Willy Wonka, Dr. Badger. So if you eat excessive carbs, more than you can utilize, they make fat, as we all know. The only place we can store that as glycogen, carbohydrates, the liver and skeletal muscle. That's your emergency supply. That's to take care of fight or flight, a unit we'll be talking about. That's where it all comes from. And we use it and we replenish it right away. And they have myoglobin, which is similar but not identical to hemoglobin, based on its utilization curve, the dissociation curve, which you're familiar with, which we go over again in blood. And the organelles are modified. There's a ton of mitochondria that have a lot of energy production, okay? But now we have within that cell the myofibrils, which within contain the filaments. We have a sarcoplasmic reticulum, which is smooth endoplasmic reticulum. There effectively is no rough. And invaginations, indentations that carry this excitability signal along that sarcolemma, along the actual plasma membrane of the muscle fiber or cell, work by way of called transverse tubules. I'm talking about them. Thankfully, the animations show them to you very well. How does that signal get deep inside? How do we take a signal from out here and get it down into here? And how does it get around all of these individual fibers? You're going to see that, how that signal works. And then we get into the physiology, and you're going to get sick and tired of hearing about ligand-gated and voltage-gated channels. Believe me. You don't have to thank me. Why do I try to be humorous? Densely packed rods, single muscle fiber thousands, 80% of the volume, striated individual units called sarcomeres, the tiny filaments. We're going to look mostly at their components of actin, which you should be familiar with, and myosin, which you should be familiar with. But we'll review it again and really sort of the central anatomy is associated with it, but probably not today. I mean, we'll get more into that probably on Friday. Okay, so we're pulling here. Here's that individual. Here's the sarcolemma. We're looking at an individual fiber. We're going to pull out one of those fibrils, and now you can see closely those little blue lines. The divides on each of those fibrils effectively is a sarcomere. The little blue lines are something called Z, as in Zelma, as in zoo. Okay, Z discs, because they zigzag, molecularly speaking. And so the boundaries, I love this, boundaries of a sarcomere from Z disc to Z disc. Or as the song says, from Z to shining Z. Let's have a little patriotism here, you know. Striations. The striped appearance from these alternating light and dark bands. You can't ever get this wrong. It's like one of those things that LR6SO43 I told you about. This is A bands and I bands. Can't miss it. The A band is the dark region. Notice there's an A in dark. The I bands are light regions. There's an I in light. There may not be an I in team, but there's an I in light. Right? So my son was, my oldest was a goalie. So I said there was always an I in goalie. Just ignore those other guys. Stop the shots. Okay? So A bands and I bands, you're going to see them as we're going to see a middle area. I don't get crazy about myomycin and other protein. It's more of an anchoring protein. It looks this looks like a little H, a lighter region in the middle of the dark A band. We call it H zone just because of its shape. But the key is those zigzag or coin-shaped proteins midline of the light I band. You're going to see it. Not so well from this. This would be a typical microscope, high-power microscopic view of skeletal muscle. You get any kind of skeletal muscle. Remember, I wanted to get into this and some of the physiology of it more Friday and more Monday in lecture before we go into lab. Like I said, we're planning to have a wet lab. I just got to make sure the zoo lab is available, and we'll do the rabbit muscle lab. It's a kit. It comes from like Carolina Biological. Maybe you did it before. It's kind of fun. It doesn't take terribly long. From a report standpoint, we do it in groups. So depending, there's 15 of us. We'll probably do it in 3 different groups of 5 people. So you can just hand it in as a group assignment. Make life easy for you. Sarcomeres, smallest unit. This is and when we talk about the functional unit, it's really where the understanding comes with any of these things. It's like the osteon, you might remember, is the functional unit of bone. When we get to the kidney, we'll revisit the nephron, the functional unit of the kidney. So sometimes they're both structural and functional. This would be an example. The A band is completely contained within them, and half of an I band. That Z disc kind of has half an I band on equal sides. So there's the area between the Z discs that are there. That's the boundaries. A line end to end. Myofibril boxcars work for me. Voila. So you can see it. So here, we'll just take this arbitrarily. And this is something that's very easily identifiable when we understand it molecularly. The red material you're going to see is the myosin, and the blue is the fibrous actin. If you remember actin from the cell, from what we did in anatomy, I'm sure you've covered it. Tiny actin filaments make the cleavage furrow. Actin make microfilaments. Actin can assemble and disassemble. So it's a globular protein, which means it can dissolve in cytoplasm. When they start to string together, and I always use the analogy of a double string of pearls. It's like a string of pearls twisted like that, so it looks double. Like my mom, the ladies used to wear a lot of pearls back ages ago. And the perfume and the atomizer. A little history wouldn't hurt you. It really wouldn't. So when it strings together, it becomes F actin, not F-actin in the sense of F-actin, but fibrous actin, because now it's insoluble. We're going to look at these a lot. Proteins that are soluble, but when they string together, and none more famous than fibrinogen, which when it becomes insoluble, it's fibrin, and that's really what the clotting mechanism, that's what the clotting cascade is all about. So we go from G, globular actin, to F-actin. We're looking at F-actin. In this case, it stays that way because these are insoluble proteins. They're not designed to dissolve. When they break down, when somebody dies, they denature. And so, and you can see we have the strands of myosin in the red completely within the sarcomere, but half the strand is on either side equally of the Z disc. And this is the area that overlaps. The dark band, notwithstanding the H zone in this middle area, which is somewhat lighter, is due to overlap. You're going to look closely here. The relationship, I got my marker. The relationship is roughly 2-to-1, light, the thin filaments, which are actin, to the thicker filaments of myosin. So it looks kind of like this. Here's the myosin in the middle, and you'll see it. It's exactly there on that diagram. It'd be much better than my terrible artwork trying to draw it. But they, as you can see, they're going to share different areas like this. And now you have another thick one here, 2 over here, and you get to kind of get the idea. So the ratio is roughly 2-to-1, light, light, thin to thick. You're going to see it's 12 or 13-to-1 when we get the smooth muscle. So the overlap is there. We just can't see it. There's just a vast, larger amount of the thin ratio to the thick filaments. And here you can see there are no thick filaments unless it starts to shorten. Orderly arrangement, thin filaments extend across the I band, partway into the A band. You're going to see what happens is they get closer, backing up. When it contracts, you're going to see these are going to get closer to the midline. So they have they have an endpoint, an anatomical endpoint. So each muscle can only contract so much. Just each muscle fiber can only contract so much. The myosin filaments, which are thick, extend the length of the A band, and they're connected at the M line. They're tethered mostly to the M line, but they're also attached to the Z disc. Uniquely, you're going to see the protein. It's called titin, Titin. Okay, not like Clash of the Titans or Wrath of the Titans, but titin, and it looks like a little spring. And indeed, the elasticity that we have with our muscles comes from that titin. I assume I'm pronouncing it correct. Great movie. The original Clash of the Titans from 1981 is a brilliant display of how they used to do monsters and stuff like this called stop action, where you'd have a monster would be like this, and you'd move it a smidgen, take a picture, a smidgen, thousands of pictures to make it 200. That's the way they used to do all those Godzilla and all those movies, stuff like that. And the master of all that was a guy named Ray Harryhausen, who was doing the work in the 30s, like the original King Kong. I'm an old movie buff too. I have a lot of hobbies. You get the idea? Spend too much time at home. Too much time. So, and again, you can see the hexagonal appearance. Here it is. So where it's an I band, all thin. Where it's an H zone, you can see thick, but they're wide apart. The M line, they're tethered together. But here in the A band, that's the better drawing that I have. You can see, just like I was trying to draw, they're shared. And so that really gives you an idea why. And it's really just because they overlap, light can't pass through them. I'm not going to go into anisotropic and inotropic and all that type of stuff. That Buxton guy downstairs. Ask him. He's not in a good mood. My team, Liverpool, is 1 point ahead of him in the table. Sorry. Liverpool won the championship last year, so I was a piker. So the thick filaments are composed of myosin. And again, we're going to do a lot of this composition. You all did antibodies. We're going to review them again. Heavy and light chains, long and short chains. This has 2 heavy and 4 light chains. The heavy chains form this long tail. The light chains make up a head. Okay, and you're doing contraction. This is where crossbreed, and we'll go into this in detail. So we're going to look at the interaction between the myosin heads, which you will see have an active site that attaches to the actin. The actins have an active site that attaches to the myosin. Remember, when we talked about anatomy early on, comment I made was some things are named for what bone they're on, some things where they're going. Okay, the zygomatic process on the temporal bone going to the zygomatic bone, the temporal process of the zygomatic bone going to the temporal bone. They met to form the zygomatic arch. So this is where you build a little bit more on that stuff. So myosins are offset from each other, resulting in a staggered array. This is really important anatomically, because they're constantly, you're going to see when a muscle contracts, when you have to move it a little bit versus moving it more and more and more. Let's say you're lifting something very light versus trying to lift something very heavy. You're attaching and detaching. You're going to see it in the animation. They attach, pivot, attach, detach, attach, pivot. They're elastic. If they all attach simultaneously, it would be like this, like pulling down the blind, you know, bing, because of the elasticity. So you're going to have half of them stay attached while half of them contract, and then so on. So there's always some that are attached. So when you're contracting, it's not like you have to go back to square one. It's not like I lifted that. Oh, I got to lift this now. I got to put it all the way down. No. That's because of this interesting, eccentric, irregular, or staggered arrangement that you will see. So the anatomy tells us about the function. See, once again, I will acquaint you with my teaching philosophy again because we have newbies in here. If I'm not excited, why would you be? So every time I teach this, I get excited. Jenna, you're looking, it's a tough night again. You got to stop working all those hours. You can't, we're going to have to, you probably don't drink coffee. Let me see you. Sorry, good for you. Stay away from my wife and I drink too much of it. Thin filaments composed of fibrous actin. Remember, it starts as G. The G actins bear active sites for myosin, so we call them myosin active sites, even though they're on actin, versus actin active sites, even though they're on myosin. Names are another way to get tripped up on a test. The G actin units become F filamentous or fibrous, I called it. They twist together to form a thin filament. In addition to that, when you see the illustrations, we'll go over this many times, we have what are called, those are it's all proteins. We have regulatory protein. Tropomyosin is a strand-like protein that threads through it and covers the active sites where the muscle is at rest, and it's held in place by plugs of another protein called troponin. Okay, if I was sick, I went to the emergency room and said, I've got a stomach ache, I'm having trouble coughing, my breathing make it tough. The first test they're going to run, maybe not, in a group of tests, the first thing they've got to rule out is a heart attack. Because of my age, because of my weight, because of my medical history, because of my high blood pressure. They do what's called a troponin level, called cardiac troponin. Skeletal muscle has that. When a skeletal muscle cell dies, heart attack. The troponin is released in the bloodstream. You could do the same for a skeletal muscle because they're slightly different troponins. When a skeletal muscle is injured, you would see that. We really don't have to do that. You can analyze a skeletal muscle. But in the old days, when we had to diagnose a heart attack, we had to do enzyme levels over 3 days. We had to do serial x-rays, serial EKGs, You can make the diagnosis now. Really an hour or so. Just to give you an idea. That's where troponin is important. For those of you moving forward in careers that involve medicine, troponin, something we didn't have. So they're regulatory proteins that are associated with the actin. This is the thick filament. The thick filament is twisted there years ago, I was Dr. Badger. I would bring in candy. I mean, if that's not a Twizzler, I don't know what is. Right? Is that a Twizzler or what? The trouble is, I just eat them. And my wife is in Good and Plenty Anonymous. Really? I'm in Mike and Ike Anonymous. I'm Lou. I have not been sober for Mike and Ike's ever. So you can see the binding sites to actin are there. So they call them actin binding sites. Those are the globular portions. Those are the lighter chains, the heavier chains. And you can see they're staggered, making this giant myosin strand is made of a bunch of little strands that are their individual strands. And you can see the offset of the heads. And normally you're going to find them, important when we get into the physiology of the cocked position. That's the ready position. They're always ready to contract. We preload them with energy. We do that for two reasons. One, they're ready to contract, so it's convenient. Two, it's the only way we can get them to detach from when they previously contracted. We're going to explore how ATP plays that role. And again, the animations do a great job. Here's that double string of pearls configuration. The active sites, the binding sites for myosin, are covered by the tropomyosin. And so, like a push pin or a thumbtack, any analogy will work. Whatever works for you, the troponin holds it in place. So, couldn't I have done this in advance? What a doofus. So it's like, I'm a doofus. The dog is more of a doofus than I am. He won't shovel. So it's like they're covering those sites, and what holds them in place, right, are the troponin, the little pins. But if those move out of the way, boom, they're exposed. Again, I used to do that with not the regular Twizzlers, but the peel apart Twizzlers, which may even be better. It's a candy thing. It's Anthony Newley and Leslie Bracus. Candyman, that song. Candyman can. Right? Listen to the Sammy Davis Jr. version. It was a #1 hit. I know, 6,000 years ago, before any of you were even conceived. So, other proteins help. Here's the titin. You can see its spring-like configuration. It holds the thick filaments. It attaches them, so they're not attached directly, but indirectly to the Z disc. Helps recoil, resist excessive stretching. Dystrophin is named for the disease, muscular dystrophy. DYS just means irregularity. Troph means growth. This is when you learned a bit, I'm sure, in genetics, many of you, about, and I know forgive me, Dr. Hillwood, I call it Duchenne's, not Duquesne. Duquesne's a university. My son went there, got his master's. Duchenne's muscular dystrophy links the thin filaments, so this dystrophin links the thin filaments to the proteins of the sarcolemma. And there's other ones that are there. You don't have to worry. So Duchenne, not Duquesne, is the most common. We're all familiar with it. Sex-linked, recessive, so it's almost exclusively males. I'm not going to go into the genetics of it. What happens is, what a dystrophy does is the muscle doesn't form, and when the muscle doesn't form, inevitably it's replaced by fat. And it gives parents a misconception. You see these types, oh, look at those good-sized legs, and arms that this little boy has, and it turns out they're not muscle. They're not going to turn into muscle. They begin to deteriorate. And certainly the big problem is breathing, and that's related. And it goes from the extremities on upward, as you can see, and eventually you're going to end up with heart, respiratory problems. And I mean, the lifespan is increasing considerably over time, just to let you know. So we really have improved. There was, for years on Labor Day, they had a muscular dystrophy telethon that went for years, 24 hours on Labor Day, wasted in those days a ton of money. Okay, now money raising is done, but that was Labor Day weekend, where basically everybody had that Monday, so it ended like the Monday Labor Day, let's say 6 o'clock in the evening. and started the Sunday before 6 o'clock. The late great comedian Jerry Lewis, that was his pet charity, and he stayed awake for the whole time, did anything to make money with regard to it. So they named the gene for the protein. Okay, the protein for the genetic defect, really. Okay, extracellular matrix. Okay, tears easily, all sorts of bad stuff, inflammation, apoptosis, and then it's replaced by by fatty tissue and scar tissue, etc. You can see that here. So, I mean, there's been some improvements, I think, over the time. I don't know therapeutically where it sits now. We will start with the sarcoplasmic reticulum and begin to understand the A bands, the I bands, T tubules, the triads. There's a lot of terminology before we really get into the nitty gritty of how this all moves. Thank you. All the stuff is here. Listen to that one I did yesterday because we were too busy shoveling. I was exhausted after that. Thank you. Bye bye.